What happens next after successful fertilization or conception? So we call it development of zygote. So definition: a study of which of a known as embryology or embryo development. Okay. Okay. The key point here: series of mitotic division. We call it cleavage. So this is the definition: a division of cells by mitosis with no significant growth. Okay. No significant growth here, talking about the diameter of this conceptus. Okay. The cell is increased in number, the cell proliferate, but the diameter of this conceptus is still the same. Okay. If I want to draw here, where is it? Okay. Let's see. Bayangkan ni adalah suatu diameter yang sama sahaja. Tak pandai pula saya nak buat ni. Okay. So, from one cell, ada ni. Only one cell. So, it will divide into two. So, it will divide into two. 1 and 2. And then it will divide into 4. Okay, 1, 4. Or just draw it here. 4. Okay. And then further divide by 8. Okay, so the cell increase in number but no significant growth. The diameter of this conceptus or even the size of this conceptus is still, it will stay the same until blastulation or until the next stage. Okay. After fertilization, embryo development proceed through this one: cleavage, blastulation, and organogenesis. If you remember my slide, we have another one, blastulation. Okay, because under references any embryo development book, okay, there are another step. So you might want to find the definition of embryo, the blastulation. So blastulation is compaction of cells to become a hollow ball here. Okay. Important events regulating development. Okay, so this is an important event to develop to develop the animal's body. Okay. This diagram here, cleavage. So you can see the same, no significant size growth inside, size growth. Okay. 1 cells, 2 cells, 4 cells, 8 cells, and then blastula. So this one, blastula, blastocyst, hollow ball, is actually compaction of the cells just now. And then we cut across this blastula, blastocele. We can see blastosole is actually the space of this hollow ball, blastosole. And granulation is the next stage, and then next stage is organogenesis. Okay, blastulation, granulation is like a invagination of the cells towards inner. So therefore, when this happens, the invagination of the cells towards inner, it will produce 3 layers of tissue that eventually contribute to organogenesis, meaning this tissue will develop and differentiate into different parts of the body system, organ. Okay. All right, next. Okay, so you may use this slide here and you add your own notes, the elaboration of each steps. Okay. Okay. This is fertilization, and then fertilization successful. Now we call it conceptus. Okay, and then you can see 1 cell, 2 cell, 4 cell, and so on. So cleavage, and then blastulation, compaction. Okay, and we have blastocyst, a hollow ball, And then granulation, tak ada kat sini. Okay. Yeah, and then granulation, when we can observe the three layers of the tissue. Okay. Okay, I think okay lah ni sama je. I mean berulang-ulang ya. Morula, bastula. Okay, next. Ni dah cakap sama. Okay, I think the new terms here, blastomeres. Okay, what is the blastomeres? The smaller cells. The cells here. Okay, so 4 blastomeres, 16 or 32 or 100 blastomeres. Okay, cleavage partitions the cytoplasm of one large cells. So basically the division or proliferate of the cell is like segregate the cytoplasm from one cells. Okay, it segregate or partition the bahagi-bahagikan cytoplasm tu kepada many cells. So into many smaller cells we call it blastomeres. So blastomeres is actually this one, 1, 2, 3, 4 or 1, 2, 3, 4, 5 or many cells. Okay. Okay. So this one ada dalam buku. How the real embryonic cleavage stage. Okay. Alright. I think this G and H is quite interesting. So this one G and H is actually when this cell already cleavage and then blastulation at some stage here, these cells breaks out from the fertilization envelope. If you remember yesterday, I did talk about fertilization envelope where the zona pellucida or the perivitelline space become hardening, right? Harden, and then that layers, the zona pellucida hardening, we call it fertilization envelope. Okay, so this concept is at the end stage of cleavage, if I'm not mistaken, or blastulation, okay, it will break through from that fertilization envelope. This is how it looks like. Okay, yeah. Okay. All right. Morula, blastocyst. Okay. Okay, so this is about timing, developmental stage from the start of cleavage until 9th week. The embryo first undergoes division. Okay, ni dah tahu. Embryo, okay. 16, embryo floats free in the uterus temporarily and the uterine secretion are used for nourishment. So this one, the last 2 points here. So it will floating freely. Okay, from here until here, this stage here. The uterine wall here, the master cell, the uterine endometrium here. Each cells of this uterine wall will produce substances or nutrients that is compulsory for, that is will nourish this conceptus. Okay, until this conceptus floating freely, it will attach to this upper part of uterine wall to implant itself into this wall. Okay. Okay, okay. So next stage is the blastulation. Okay, or we call it blastocyst. How to detect which part is which stage, okay. So when we can observe a hollow or blastocele or blastocyst cavity here, we can say that is a blastocyst stage. Okay, blastulation already occur, compaction of the cells. It will produce this blastocyst. The significant feature of this blastocyst is actually we have trophoblasts, the outer layer here, and we have inner cell mass. Or maybe sometimes I just draw it ICM. Okay, inner cell mass, and then we have the cavity, the blastosome. Okay, blastocyst is a hollow ball with an inner cell mass. So each of these significant structure have their own functions. Okay. And this blastocyst, the hollow ball-like circle of cells begin at about 100 cell stage. Okay, if we look back at this cleavage, so about 100 cell stage, the compaction already occurring. Then we can observe this structure here with a hollow cavity and a compact cells at one part. Okay, and then another significant structure is the trophoblast. Okay, functioning secretes human chorionic gonadotropin, HCG. Okay, HCG is secreted by this trophoblast. Okay, this trophoblast able to secrete HCG. HCG, human chorionic gonadotropins, is essential or important to further stimulate the corpus luteum to continue producing progesterone. So in terms of timing, if you remember, we have ovulation, and then we have this day 15, 16 until day 28, we have corpus luteum, right? But the end of corpus luteum will degrade towards the end part of menstrual cycle. This ovarian cycle or uterine cycle, ovarian cycle, okay? Okay, we have corpus luteum at day 16 until 20, 21. Okay, so this corpus luteum function to produce progesterone. Okay, just revise back. Progesterone, eventually it will be peak, but it will drop towards the end of uterine cycle or ovarian cycle because the corpus luteum itself is degrading. Okay, so at the end part or at the end phase of this cycle, in order to maintain the progesterone level, okay, the trophoblast will secrete HCG. Okay, so when HCG is present, corpus luteum will continue their integrity of the cells, and so that they can still produce progesterone. Okay, so you can imagine ovulation occur, so the conception of fertilization should be occurring at this timing or period. Okay, tak boleh lah lebih, memang tak. So it's very complement time for fertilization occur for the couple that have intention to get a baby. For them to overlap the sperm and the egg viability time. Okay, so the fertilization will occur around after ovulation or around this time. So you can see if fertilization success, the cleavage stage will occur during this stage, will occur at this period of time. Okay, so towards the end of the ovarian cycle over here, okay, or yeah, the implantation should occur, okay, for a successful pregnancy. In order to determine a successful pregnancy, the endometrium need to be fully thickened until the end, right? So in order to maintain the thickening of this endometrium, the progesterone is needed, right? So the progesterone need to be at high level all the time. But corpus luteum integrity cells, it's degrading towards this end of time. So when this cleavage or the conceptus is present at this time of this period, okay, so trophoblast already present, trophoblast will secrete HCG. HCG will continue or will stimulate this corpus luteum to continue produce progesterone. Okay, therefore the blastocyst maintain the endometrium thickening, And then the blastocyst able to implant itself to the wall of the uterus by day 14. Ini kira balik lah ni. Ni hari kosong, hari 14. Okay. So kat towards the end ni dia nak implant. Okay. Excuse me. And then next after blastulation is gastrulation. So the significant features for gastrulation or when gynae, okay, when gynae or the embryologist observe under microscope at the cell stage or the embryology stage, they need to detect how they recognize that this cell or this fetus or embryo already at which stage. Okay, so the embryologist will recognize the invagination of the cells into towards the end. Okay, wording dia ni I'm afraid if my pronunciation is not so good, but I can spell it out for you. Invaginate. Okay, invagination. Invaginate the cells here, the cell structure, this one, the outer layer here, invaginate towards inner. Okay, so that's how embryologists detect this embryo is now at gastrulation, because they can observe the invagination of the layers inside. Okay. Okay. So this also a starting point of morphogenetic. What is morpho? Morpho is like a physical morphology. So it's a like a starting point to develop the structure. Okay, rearrangement of the cells of blastula into 3-layered embryo. Okay, so this rearrangement of the cell is actually the invagination of the cells, of the blastocyst cells. Okay, remember blastocyst has blastosole, the cavity, and also they have the inner cell mass. So and then the invagination had occur. Invagination actually push the cells, rearrange the cells towards inner. Okay, so that rearrangement will appear a primitive structure, primitive gut or primitive apa ni? Notochord. Okay, notochord primitive gut. Saya nak cari apa word dia, saya tak ingat lah. Baca tak ingat. Sekejap ya. Mana dia? Ting. Primitive gut, okay? Apa panggil dia ni? Mana tak tulis kat sini? Begin with the production of family germ cells by this disc, a 3-layer embryo. What else? This, something with this, primitive disc. Tak apalah, nanti you try to find the word here. The embryologist or the scientist also can detect the disc. Okay, disc. Okay, primitive gut. Okay. Okay, rearrangement of the cells of blastula into 3 layers. Okay, but it will start from 2 layers. Begin with the production of primary germ cells. Okay, invaginate, it will produce bilayer disc. Okay, bilayer disc. And then eventually will produce the 3 layers embryo. Bilayer disc is actually this one. Bilayer disc. This one. Epiblast, hypoblast. Bilayer disc. Probably not mistaken. Okay, we have the outer and another this one and this one. Okay, bilayer disc. And then eventually this bilayer disc will produce 3 layers. Ni bilayer lagi. Okay, ada epiblast ni ah. Ni ya. The layers. Embryonic cells, yolk sac, hypoblast, yeah, bilayer disc. Okay, eventually this bilayer will produce yeah, 3 layers of embryonic germ cells: ectoderm, mesoderm, and endoderm.